Thank you for that introduction, and thank you to CNN News and to ACS for this awesome honor. It's very humbling. So today I'm going to tell you about just kind of an overview of the things that we do in my lab, and we really do bridge using analytical mass spec techniques to study proteomes of diseases, and Alzheimer's disease is our most popular. So this journey began in graduate school, where I went into my PhD advisor, David Clemmer's laboratory at the time, and I told him, well, med school is off the table for me, so since I'm here at grad school, I want to do whatever it takes so that by the time I finish grad school, I can either start my own cosmetics company or I can go into L'Oreal, and I need a six-figure salary to do that. So you need to tell me what I need to do as a grad student. And what he said was, I can't guarantee that, but what I can do is put you on this interesting problem studying aging and fruit flies, and maybe something that you discover will be useful with regards to cosmetics. Didn't work out that way. However, what I did learn was a lot about technology. And so I really worked at the heart of using in-house custom-built ion mobility spectrometry, combining it with mass spec techniques to study proteomes of flies. And this was new in the laboratory, and it hadn't been done before. But what came out of it was some really interesting things. And so one of the things we were able to identify was this protein in the immune system of flies, that when you got rid of this protein, propionyl oxidase, the lifespan of the flies changed dramatically, and when you overexpressed it, the lifespan of the flies increased. So I was very drawn by this bridge of technology with important problems such as aging. So I went to the University of Kentucky and worked with Alan Butterfield, who is just a renowned expert in the world of Alzheimer's disease and redox proteomics. And what I learned from him was a lot about how to take animal model systems and get information out of them that is very relevant for human samples. So I went from working with flies to working with mice and rats and even precious human tissue in Alzheimer's disease. And I think one of the most important things that Alan was able to do for me as a postdoc was to give me this pep talk about how my role was as a chemist. And what he would do is he would sit us down in the lab and he would tell us how important the work was that we were doing in the lab. And he put it into context like this. So when you think about Alzheimer's disease, it's something that hopefully many of you are very familiar with. And currently there are 5 million people that are suffering from Alzheimer's disease. So this is the 6th leading cause of death, and 1 out of 3 seniors are going to die with AD or dementia related. And so currently the cost for Alzheimer's disease is over $220 billion. So it actually wreaks a lot of financial and economic havoc on our society. And age is the biggest risk factor. So if you look at what will happen over the next 30 years, approximately 15 million people will be affected by Alzheimer's disease. And this is a disease without a cure. And you may know some of these popular people here who have been affected by Alzheimer's disease, but myself and many people can start to fill in personal folks that we know who are suffering by this disease. And so that's what really motivates me and my lab to do the things that we do that are very complicated, is realizing that as chemists, we can actually play a role in helping to alleviate some of the suffering that people have from this disease. And so the way that we do that is we use a combination of analytical technologies to study proteins. And this is just an overview of shotgun proteomics. And the idea here is that we can start with tissue, fluids, cells, or even whole organisms that come from different model systems. And we're going to use different techniques to extract proteins out of these systems and put them into smaller pieces called peptides that we can handle a lot easier. Now, because of the complexity of the human proteome or the mouse proteome, here we're dealing with millions of components. And so we really rely on multidimensional separation tools that are either in the condensed phase or the gas phase in order to separate these peptides out before we introduce them into the mass spectrometer. And once they're inside of the mass spectrometer, we get information about intact peptide masses, but then we go on further to fragment these peptides into smaller pieces and get amino acid sequencing information. And so all of this works beautifully when you're able to carry out in the lab, and you have, in combination with the analytical techniques, bioinformatics platforms in order to help you go back to identifying what were the original things that you identified in these mixtures. So this is at the heart of what we do in the lab. And our motivation in terms of developing technology really has to do with developing a toolkit that allows us to probe many proteins at one time in the context of many different conditions. So if you think about getting to better clinical designs and facilitating experimental designs easily, how might you do that using this technology? Well, the idea that we have in the lab is to, instead of looking one protein at a time or one condition at a time, is to be able to develop technology that allows us to multiplex and look at many sample conditions simultaneously. And this ultimately allows us to increase sample throughput, to minimize the total amount of sample handling. It greatly can reduce experimental cost. And we do this in such a way that we can allow compatibility with samples that come from any type of origin. And we work on flexibility. So we have the ability to look at different types of post-translational modifications, many of which I won't be able to tell you about today. So just as an example of how we got this started in the lab, we were interested in a modification called 3-nitrotyrosine that is a result of oxidative stress in cells. And we realized that we could couple some acetylation, isotopic labeling chemistry, with some isobaric tagging chemistry, which I'll tell you about in a second. But what that allowed us to do was to be able to look for these post-translational modifications out of tissues. But the other thing that we realized along the way was that we can combine 12 or 16 samples into one single pot analysis. And so while we were looking for this for one specific post-translational modification, we've since worked on developing the technology to look for different types of modifications and just look at global levels of proteins. So this is just an example of the technology that we developed in the lab, where we combine precursor isotopic labeling with isobaric tagging. So if you envision having 12 different sample conditions, these can come from biological replicates of a wild-type animal and an Alzheimer's disease animal. What we would do is extract proteins from those samples. And so to the 6 set of samples over here, we create peptides, and then we perform a dimethylation reaction. And we do this in such a way that by altering pH, we can make it very specific to N-terminal means of peptides. And then what that allows us to do is to leave lysine residues available for chemistry. And so then we incorporate these isobaric tagging reagents. And you can buy these in kits of 6, nowadays up to 10. And so we have 2 tags, an isotopic tag on the N-terminal amine, and then the isobaric tag on the lysine amine. And so we have 6 samples here that are tagged uniquely with these isobaric tags 126 to 131, and a separate 6 samples that we tag using a heavy dimethyl group, where we replace the N-terminus with these isotopic reagents, and then we have the lysines that are also tagged with the other half of the isobaric tagging kits. So what this allows us to do is to mix 12, nowadays up to 24 samples in a one pot, and what we get back in the mass spectrometer are 3 readouts. We can look at intact peptide masses that are shifted in mass by the number of isotopic labels here. Once we isolate individual peptides and fragment them, we get the sequencing information to identify what they are. And then once we isolate an intense MS-MS fragment, we can get back reporter ions. And basically, these reporter ions just report back on the relative intensities of peptides in these individual 6 or 12 samples that we initially started with. So we've greatly increased our sample throughput. And the type of information that we get is too vast for me to really cover in 12 minutes. And so I just wanted to give you an example of what we're able to do. So we can take liver tissue that comes from Alzheimer's disease animal, and in a single shot, what we're able to look at is the relative ratios of how protein changes in disease relative to wild type. And using statistics, these things here in the red are about 60 to 100 some odd proteins that are different in the context of the disease. And when you look at how well we can sample using the combination of separations with mass spec, low abundant proteins, then we're able to look over 4 to 5 orders of magnitude in terms of the concentration differences of these proteins. And we take all this information and use a lot of sophisticated tools to look at protein pathways. But what we're able to identify is that outside of the brain, in a tissue like liver, there are many different pathways related to glucose metabolism, fatty acid metabolism, that are all also changing in the context of Alzheimer's disease as well. And so Chad Merkin, I think, had some really good advice to share this morning. When we first started working in this area, we were really up against a wall, because most of the thinking here is this is a neurodegenerative disorder, so you should be looking in the brain. However, we had the thinking that there are also other changes that are taking place in the periphery, and really wanting to understand what the role is there. And it hasn't been until the last few years that granting agencies for grants, NIH in particular, has started to recognize that there's some important work to be done there. And so this is what my group does, is we look at system-wide changes in Alzheimer's disease with disease progression. And because we have all these variables, it really requires this multiplexing technology. So I just wanted to leave you with some final thoughts on where I think not only we're headed, but where the field is headed as a whole. And so when you think about looking at individual proteins or individual post-translational modifications, there's a lot of information that you can glean there. And from a traditional biology point of view, typically you would go in a priori with some knowledge about which protein you're interested in, and you would study one protein at a time, and you would follow it down a specific pathway. And hopefully the goal is that it leads to something important that you can use therapeutically, but oftentimes it doesn't necessarily lead to that. But it helps further your understanding of disease. And so what we like to do with our technology is to take a step back from looking not just at individual proteins and peptides and pathways, but to really looking at the bigger picture of everything that's taking place in the proteome. And we think that the technology, has a lot of evolution that's taken place over the years, but really we're getting to the point where we can look at whole proteomes. We can look at tens of thousands of proteins within hours with this technology. And so what we like to do is really use this to get a comprehensive picture that allows us to better understand disease, that not only gives us insight about the disease understanding, but it actually leads to information about biomarkers that can be used in the clinic and routes for therapeutic intervention. So I think the biggest thing with our field in proteomics is that we can go from now being able to say we may be able to get disease understanding or we may lead to therapeutic interventions, to within the next 10 to 20 years really being able to actually get disease understanding and identify targets that will be useful in the context of disease. So with that, I just wanted to leave you with some thank yous to my group. So we have a very active and growing group. We went out on the 3 rivers of Pittsburgh over the summer. And so I'd just like to acknowledge my group for all the hard work that they put in and the supporting cast that I have in my family, our funders, and our collaborators. And again, thank you for this awesome opportunity to give you highlights on what we do. Thank you.